All right, now we're going to talk about the process of infection. We're going to talk about kind of what a pathogen has to do in order to gain access to our bodies and to create disease, infectious disease. So to establish an infection, a pathogen has to basically complete 5 tasks, right? So it has to be able to enter the host, it has to be able to attach to the host tissue, invade the host tissue, and obtain nutrients. All the while, it has to navigate around and fend off the immune responses, and then it can cause the disease state, and then it needs to exit the host. It has to be able to transmit to a new host. So let's talk about this first one. All right, so step 1 in becoming established. So the portal of entry is what we call this. All right, so that's any site that the pathogen basically uses to gain access to the host or to enter into the host. So because mucous membranes basically line every entrance of the body, they typically are kind of the key portals of entry into the human body. And these mucous membranes typically have specialized defenses to kind of protect against pathogen entry. The successful pathogens who are able to kind of get past this defense mechanism typically have certain virulence factors that allow them to overcome those barriers. So even though the portal of entry may be the area where the pathogen gains access, kind of the initial site, it's not necessarily where the infectious disease may develop. So it can develop in different areas as well. So the portal of entry is often determined by kind of how the pathogen is transmitted. So we have what's called the fecal oral route, where a pathogen could be shed in the stool and then enters the mouth of the next host. I know this sounds really gross, but if you think about it, it could come from shed in the stool of an animal, and you could contact it through eggs or milk or water, all right, that these pathogens could be found in. It could be arthropod borne, could be from mosquitoes or ticks, it could be sexual transmission. So these are all different ways in which some of these pathogens can gain access to our bodies. So if we talk about endogenous and exogenous, all right, so exogenous, the organisms definitely originate from outside the body and are making their way inside, versus endogenous are organisms that originate from somewhere within the same host. So an endogenous pathogen. All right, so they could already be inside, and so the bacteria is basically kind of misplaced. So it could be kind of what we talked about with opportunistic pathogens, such as a yeast infection. All right. So exogenous sources can come from contaminated food, can come from medical equipment, soil, water, contact with other animals, and of course contact with other humans can provide us with exogenous sources of pathogens. All right. Now, it says the majority have a specific, so the majority of these pathogens have a specific portal of entry. So as it says, one that provides a favorable habitat. So this is going to be an easy access for them. And it says if it goes to the wrong portal, it's not necessarily infectious, right? So if it goes into the wrong portal, a lot of times nothing's going to happen, all right? There's not more, nothing further results from it. So we can talk about Bacillus anthracis. which can enter the skin through what's called cutaneous anthrax. So anthracis refers to anthrax. So Bacillus anthracis is the bacteria that causes anthrax, and it can enter through the skin and cause cutaneous anthrax, or it could enter through your respiratory tract or gain access into your gastrointestinal tract if you were to ingest it. So there's several different portals of entry for Bacillus anthracis, and depending upon where it enters, it can have different virulences. So cutaneous anthrax has a very low mortality rate compared to if it were to enter through your respiratory or your gastrointestinal tract. So as we know, as it says there, pathogens can have more than one portal, and because of that, there can be different virulences based on that. So for pathogens to establish disease, they first have to infect the host. And we have what's called the infectious dose, which describes how many cells or virions are needed to establish an infection. So the infectious dose, or the ID, is the minimum number of microbes necessary to cause an infection. And this is going to vary depending upon the pathogen. So the more infectious the pathogen, the lower the infectious dose. So we have different examples here. So it says the infectious dose for Q fever is just a single cell. So Q fever is something that you can contract by coming in contact with urine or feces or milk from goats or cattle. And so this is just a single cell can cause Q fever. Whereas you see the infectious dose for typhoid fever is up to 10,000 cells. So you need a lot more to cause the infectious disease. Now, fortunately, just because a pathogen is highly infectious doesn't mean that it is especially dangerous, right? So cold viruses are highly infectious. We don't need a lot of cells, but they're not usually deadly, okay? So the more infectious the pathogen, the lower the infectious dose. But once again, even though it's highly infectious doesn't mean that it's necessarily dangerous. And I don't expect you to remember these infectious dose numbers for these different infectious diseases. That's just an example there. All right, so becoming established, attaching to the host. So right, once they've gotten inside, now they need to attach to the host cells. So they need to adhere to the host cells, right? So they have to be able to bind to the cells so that they're not flushed from the body at all, right? So binding can take place with several different mechanisms that we've already talked about. So fimbriae, we mentioned, are those extensions that occur off the top of the cell that allow them to attach to other types of cells. Surface proteins can allow them to bind to other types of receptor protein molecules on host cells. So there's that mechanism. Or if we're talking about something larger, we've got parasitic worms. They've got hooks, they've got barbs, they've got suckers, which they can use to attach to the host. All right, so there's a wide variety of different mechanisms that they use. A lot of times it's just simply, the surface proteins kind of tricking the cell and using a certain type of molecule on the outside of their cell that will bind to a receptor on the host cell and saying, okay, I'm something that you normally bind to, so come on, let's make a connection. Now, once the pathogen has bound to the host cell, several different things can happen. It can stay on the surface of the host cell, or it might pass through between cells and invade deeper inside the tissue, or it can get inside of the host cell and just kind of hang out inside of the cell. Now, once they've accomplished this invasion, they can damage host tissues and generate a lot of different side effects. So, step 3, all right, so once they've become established, your body's going, okay, something's going on here. I detect some invaders. So these pathogens have to be able to avoid the immune response, right? So surviving the host defenses. And they have to do this so that they can safely replicate inside your body. Now, the body has evolved many different defenses to prevent and overcome pathogen infections. And of course, pathogens have evolved countermeasures to escape your host defenses. So it's very common for pathogens to employ multiple strategies to avoid detection. All right, so a lot of times your immune, to avoid your immune response, pathogens might hide in a biofilm. They might enter a latent state, kind of a dormant state. They might change what's called antigenic factors to trick the immune system into thinking that it's just a harmless body cell because of what it has on the outside of it. So these are all things that these pathogens can kind of employ. So even if the pathogen can't avoid immune detection, it can kind of limit what the immune system can do. So one thing that it does is it can interfere with phagocytosis. So certain immune system cells are called phagocytes, and their job is to engulf and destroy pathogens. And so it's kind of like the amoebas that we saw, where they can extend outward and kind of engulf whole cells. So some pathogens avoid phagocytosis by making a capsule or having that slime layer around them that kind of makes them undigestible. And even if they are digested, it kind of blocks the fusion of vesicles that have digestive enzymes inside of them, so that those vesicles cannot fuse with the vesicle that has the pathogen inside of it. And so that keeps the pathogen whole inside of that phagocyte. They also can produce leukocidins, which says that are toxins that kill the phagocytes. So leukocidins a lot of times will cause pores to develop in the membranes of the cells, and that causes them to die or to kind of burst because of that. So leukocidins are things that the pathogens secrete to basically counter your immune cells. All right, so, and then I said we've got the slime capsules that make them undigestible, and then they're also able to survive inside of a host cell. So we said that some will penetrate deeper into the tissue, pathogens may hang out on the outside of the cell, and some pathogens may actually enter into the cell. So if they enter into the cell, it's very hard for your immune system to detect those pathogens once they're inside of a cell. So they avoid phagocytosis simply because they're residing inside of the host cell. All right, so some bacteria are able to secrete enzymes into their immediate environment, and these are called exoenzymes because they're going outside. They're being secreted to the outside of the pathogen's body. And these are part of their normal metabolic